So I would like to start with thanking the organizers and most heartedly Professors Ivanov and Gabibov for the flattering invitation, and I will try to use this opportunity to update the distinguished audience on our progress in toxin research. I would like to drive your attention to our lab logo here. And you might spot the neuron. So we are interested in how the nervous system functions. And you might also recognize in the center the beast from the previous talk. So we're using natural toxins to manipulate the function of the nervous system. People are usually afraid of poisonous and venomous creatures. Let's consider this example here, this red beast. It looks quite pretty to me, but I have arachnophobic friends who would scream and run away on just spotting this image. This spider inhabits Central Africa, and local tribes are scared of this spider, literally as hell. And perhaps it is due to co-evolution of primates and spiders, is that modern people have inherent fear of these creatures. This is not a well-documented theory, so I put a smiling face down there, which is also a way of saying hello to a subset of our online audience today. This fear is not groundless, and here's a video which I would not start, showing to you the effects, presenting the effects of this spider venom in mice, and just trust me that the fate of those mice is miserable. But the question that I would like to pose is, are toxins only villains, or can they play good guys? And I will try to convince you that indeed they can. First, toxins bind specifically to particular targets. And here are several examples that actually constitute the legacy and pride of this institute. So toxins from scorpions, from black widows, and from Elapides snakes bind to sodium channels in the axon, to the presynaptic or transsynaptic, as we've heard just now, proteins, and postsynaptic neurotransmitter receptors. All of those types of toxin are actually lethal, but they were used by scientists to isolate just those proteins. And what we know on the molecular level of neurotransmission today is largely due to use of toxins as molecular tools. So this is one of the examples where you feel that toxins are playing a good role. Moreover, they not only interact with particular targets, but they recognize particular parts of the targets. Here's an example. This is a sodium channel, and certain toxins bind to the pore domain and interact directly with the ion flux, whereas others bind distantly to so-called voltage sensing domains to affect gating properties. So toxins can modify the function of their targets in a certain way. And here's again an example with sodium channels. So pore blockers give a simple block of the current, whereas those toxins that bind to voltage sensing domains alter the gating properties. For instance, they can enhance the activation, or they can inhibit the inactivation of the channels. Moreover, toxins most often arrive in libraries. So in just one spider venom, there are dozens or hundreds of toxins which are related, but still different. They contain absolutely conserved cysteine residues, and therefore the disulfide core, but then the loops are hypervariable. And this hypervariability leads to a variability of the targets that they bind to. Here's just a small list of toxins, of spider toxins, and you can see the targets that they affect and the functional properties that they affect. So perhaps there is actually a sort of relation between a vast variability of toxins, a continuum of toxins, and the variability of corresponding receptors. And moreover, we can boldly suggest that to any given ion channel, there is a specific toxin due to this remarkable variability. So, toxins recognize particular targets. They bind to particular parts of those targets. They affect the function of those targets in a particular way. And given the variability, you can find a specific toxin to perhaps any ion channel. What can we do about it? I will show you several examples. One is here. So this is a Rode-McKinnon structure of a potassium channel. And there you see a toxin, a pore blocker, sitting nicely in the mouth of the pore, blocking the ion flux. So what if we do this thing? What if we play this trick? What we do, we attach a fluorescent protein to the toxin. And then we have a genetically encoded fluorescent label, which presents high affinity to the target and high specificity because of the properties of the toxin. And we can use these tools to visualize the ion channels, to make them visible. This is indeed what happens. So here are some brain slices showing to you the distribution of a particular subset of potassium channels. My other examples come from the field of drug discovery. So one is about pain. It is common knowledge that venomous creatures induce pain, and hence it is obvious that they contain toxins that bind to pain receptors, those ion channels that induce pain response, and activate those pain receptors. But due to this variability that I was referring you to, we also find in venoms blockers of pain receptors. For instance, we were first to identify a highly affinity blocker of a subset of pain receptors called purinergic or P2X receptors. These are involved in several pathological pain states. They are a validated target in several pathological pain states. And this toxin from this beautiful spider up there recognizes specifically P2X3 isoform involved in pain response and blocks it with a high affinity. This molecule has has now entered the pipeline, and we'll see what comes out of it. My final example today will be a bit different in the way that the previous toxins that I was referring you to affected normal existing native ion channels. But this is an example where a natural toxin affects mutated ion channels that we find in pathologies. So a group of diseases, when ion channels are malfunctioning, are called channelopathies. And here's one example. So this disease is called hypokalemic periodic paralysis, type 2. So it is due to a mutation in the voltage-gated sodium channel muscle type. The mutations are in the voltage sensing domains, and they cause the ion channel to leak. So in a native ion channel, there is current only through the pore domain, the central pore domain, whereas in the mutant channel, we also have an aberrant current through the voltage sensing domain. And currently, there are no drugs that specifically recognize those mutated channels. But this small, tiny little spider, just a few millimeters, secretes a toxin that does the job. So what it does, it blocks this aberrant current through the mutated voltage sensing domain, as shown here. We did NMR, we did functional studies. This toxin is highly preferring the first domain. It is selective towards just one of the voltage sensing domain, a particular voltage sensing domain in the channel. And it interacts with the domain through so-called membrane catalysis. So what happens is the toxin associates with the membrane, and then perhaps by lateral diffusion, it associates with the voltage sensor and blocks this aberrant current. There are actually a number of toxins that we consider drug hits today. Some of them are quite at an advanced stage. Others are just in fundamental research. But you can see that there are several diseases that could be tackled potentially by use of toxins. I will sum up with telling you our research priorities. So I was trying to show you how we use toxins as ion channel paint. We are trying to make analgesics out of natural toxins. And we believe that certain toxins present promise in terms of combating channelopathies. I would like to acknowledge many people working primarily in this institute, but then also at different research institutions and universities of this country. Also our colleagues at KU Leuven in Belgium and University College London, England. Here are the smiling faces of our laboratory and two more smiling faces. This conference is in memory of Professor Ovchinnikov, and I hope that you certainly recognize him. And I also hope that you recognize the other smiling face, who is Professor Eugene Grishin. So Ovchinnikov initiated toxin research in this institute, among other things, and Grishin was his follower and a true champion, world champion of toxin research. And those seminal toxins that I was referring you to in one of the first slides were his achievements. I think that this picture was taken in Peru. Professor Ovchinnikova will correct me if I'm wrong. And this is where they established a biocamp with biolaboratories. And actually, one of the most, perhaps the most stunning work with ant venoms was performed on ants collected in Amazon selva in those days. With this, I would like to thank you for your attention.
Thank you, Alex. And Professor Schlesinger has a first question.
You mentioned that these toxins come in families that are very related, and you showed they're an impressive related group. So are they encoded by distinct genes, or does it involve alternative splicing or other mechanisms for diversity generation?
Current answer is a bit unstimulating. They are directly encoded, most of toxins are directly encoded in the genome, whereas other possibilities of generation of variability, such as you referred to alternative splicing or post-transcriptional modifications, are just minor.
Yeah, I have one question for you. At the end, you mentioned potential hits as multiple sclerosis and arthritis. Do you want to elaborate more on this, how this would work?
There's an ion channel, which is a validated target in autoimmune diseases, and that is Kv1.3. Yeah. Beautiful work from Professor Chandy. We have some good toxins, selective toxins, to that particular ion channels, to that particular ion channel isoform. So it is the blockers that you need in autoimmune diseases. So these channels are hyperexpressed in a subset of T cells, and you need to block them to stop the proliferation of those T cells.
Yeah, but they're not expressed in autoreactive T cells, just the T cells in general. So you can induce an immune depression, right? They have been claimed to be expressed selectively in autoreactive T cells.
Well, thank you, Alex, again.